This is your lecture for the Chapter 9 of the nervous system. All right, so basic aspects of the nervous system, it does things like sensory input, integration, processing, that's this idea that we have consciousness, we can think, and motor output. So input, sensory, that's things coming from the periphery, light, sound, touch, that kind of feedback, as well as things like chemoreceptors that are monitoring your blood CO2 and O2 levels, your blood glucose, all of that comes as part of sensory input as well. And then that motor output. So based on what you're getting in, your central nervous system will decide what type of response needs to be made, and then you have this motor output, which is the actual effector response that causes some type of change. So really, though, the nervous system is involved in thinking, movement, internal processes of physiology. It's one of the main regulating systems of the body. It uses things like neurons that are the cells. Those are the cells of the nervous system. Like in the muscle system, we have muscle fibers. We have neurons in the nervous system that are going to be using those electrical impulses to communicate. And then the support cells are the neuroglia. They also use things like neurotransmitters that are these kind of translators between the electrical signals. If you remember in lab last week, I mentioned that in the nervous system you have this electrical signal, and even though it's going to be an electrical signal coming in and it's an electrical signal going through the cell, there has to usually be some type of chemical mediator that's translating that signal. And so neurotransmitters do that. And as I said, the nervous system is involved in regulation, and it does that in conjunction with the endocrine system to maintain homeostasis. We'll learn about the endocrine system in a future chapter. So here you go. You have that sensory input, the brain and spinal cord, the central nervous system, integrating that, deciding what type of change needs to be made, if any, and then the motor effector output. So I don't know, maybe in this case you see something coming and your effector output is to jump out of the way. So we can look at the different ways the nervous system is organized. So you have the central and peripheral nervous systems. The central nervous system is your brain and spinal cord. That's all the integration, central higher decision making, conscious thought sometimes, and unconscious thought. And then the peripheral nervous system, those are all the cranial and spinal nerves. So as soon as we start getting into cranial and spinal nerves, that is part of the peripheral nervous system. They have both sensory and motor divisions. So it's not like the peripheral nervous system is just sensory. Again, you have to have the sensory input coming in, and then you have to get the output back to those cells. And so the peripheral nervous system is going to contain both these, we're going to use new words, afferent and efferent divisions. I usually like to remember sensory afferent, A arriving, so that's going towards the central nervous system, and E exiting, this is leaving the central nervous system. Functionally, we can talk about the somatic and autonomic nervous system divisions within the PNS, the peripheral nervous system. So somatic is what's going to be controlling voluntary skeletal muscles. Somatic is used a lot of times used in conjunction with the voluntary body. While as the autonomic are those things that we don't have conscious control of. Your blood pressure, your blood glucose, the way your digestive system regulates, that's all based on the autonomic nervous system. Here you can just kind of see a description of that, begin highlighting this central nervous system portion in gray, and then all the yellow peripheral nerves. And then you can see sensory input, afferent arriving to the central nervous system, efferent exiting signals. Those might be somatic nervous system signals to move your skeletal muscle, or autonomic nervous systems to change some type of blood chemistry. So breaking down these functions a little bit more, the sensory function is used from receptors. Remember, we learned about those tactile receptors, free nerve endings, all of those things in our integumentary lab portion. So those are all types of receptors that are detecting internal or external changes, and then they're taking that information into the central nervous system. Once there, you have interneurons, what we call those interneurons, that have this integrative function to then process whatever that sensory input is. Is it that, there's lights being turned on, we're getting a more excitatory signals from that optic nerve or from those optic receptors. Okay, we need to change some type of dilation or contraction in the pupil. That's going to be this motor output. So this is the effector portion. Effectors are muscles or glands that respond to decisions made in the CNS. So based on this integrative function, your effectors are then going to change some output. Looking at the structure of a neuron, we can see there's a lot of times some very similarity, a lot of similarities in these, even though there are functional differences. So neurons are going to have the soma, the cell body. That's going to have a lot of those things like mitochondria, stuff for synthesizing proteins and different neurotransmitters, as well as a nucleus. They're also going to have dendrites. Dendrites, dender is Latin for tree, and so they look like these little branches that are coming off of the cell body, and dendrites are receptive surfaces. So dendrites are what are receiving the receiving portion of a neuron, where receiving it from some other neuron structure. And then the axon away. This is taking impulses away from the cell body based on whether this signal from the dendrites coming in, whether it's excitatory or inhibitory, it's going to determine what type of signal is sent from the axon. Only one axon in each neuron. We'll see there are different types of neurons, but they all only have one axon carrying one signal away. Neurons may also have a myelin sheath. This is not all of them, but some of them do. Myelin is this fatty sheath that covers large axons. They're called the myelinated fibers. And in those myelinated fibers, they have gaps. So you can kind of think like if you're watching this obviously on a device, maybe you have a cord nearby you, a power cord of some kind potentially. You can notice that it's wrapped in a coating. It's wrapped in insulation. And when you see wires that don't have insulation, where the actual metal bits are sticking out, you know instinctively, or hopefully you know, oh, I shouldn't touch that. That's dangerous. That could be dangerous because that signal, that electricity could get out. It could shock me. It could start a fire. It could be dangerous. So myelin acts the same kind of way. It's going to insulate the axons of neurons to help trap that electrical signal, or rather to help insulate that portion of the axon so that the signal can jump. So this is what this myelin sheath increases the conduction speed of nerve impulses. It's because by having this myelinated sheath, or an incomplete myelination, it allows the signal to hop over those unmyelinated portions of the axon. We'll see that in a moment. Those unmyelinated portions are called the nodes of Ranvier. So here you can see this yellow myelination, because it is fat, so it's yellow, and you have these gaps, these nodes of Ranvier. And so the signal, it does not pass under the myelination, it hops over these little sections that are unmyelinated. And that's how the signal can then be a lot faster versus having to pass along every portion of the axon. You can also see just some basic structures of this in here. A neuron structure is very similar to a muscle structure, so it's filled with these neurofibrils as it gets smaller and smaller. It's wrapped in the myelin sheath, this lamellated structure. You can see this thing also called the neurolemma. We'll see that that's only in the peripheral nervous system. This is what allows the peripheral nervous system nerves to heal. So if you ever have nerve damage in the peripheral nervous system, not ever, but depending on the type of damage, a lot of times it can still heal itself. Central nervous system is not as effective in that. Okay, so looking at structural classification within neurons, size, shape, and structure is going to determine their classification. So multipolar neurons, bipolar, and unipolar, that should kind of key you into what they're going to look like. So unipolar have one. These are sensory neurons. Typically, you'll find those in the peripheral nervous system because they're sensory, right? Bipolar are going to have two processes coming from the body. These are a lot of times in special senses. And then multipolar have many dendrites and then one axon. So here you go. You can see those. This multipolar having all of these dendrites attached to it. And again, think about dendrites, dender branches, trees. You can see how they're kind of twiggy looking. Functionally, we can talk about sensory, those afferent arriving neurons, interneurons within the central nervous system, and then motor effector neurons, effector exiting, that are taking out the signals. This is again just showing you the flow within neurons, how that processing of information can work. All right, the neuroglia, these are the support cells. They're helping with repair, regeneration, some of them are bacteriophages. So you have 4 in the central nervous system and 2 in the peripheral nervous system. They're doing things like filling in that spaces, structural support, no generation or conduction of nerve impulses with the neuroglia. You have microglia, oligodendrocytes, and ependymal cells. The microglia are phagocytes. So these are just a special type of white blood cell. We'll learn as we go through the different body systems that Most systems are going to have, or all systems are going to have, some type of phagocyte, some type of white blood cell embedded within them or in that tissue, and each of those are going to have a specific name. So in the nervous system, we call it microglia. Those are the white blood cells, the phagocytes of the nervous system. Oligodendrocytes, they produce the myelin sheath in the central nervous system within the brain and spinal cord. The particular cells that are secreting or that produce that myelin sheath are called oligodendrocytes. And then ependymal cells produce the cerebrospinal fluid, CSF, within the central nervous system, and we'll get more into its functions. The fourth cell in the central nervous system are astrocytes. These provide a lot of structural support. They're kind of in between blood vessels and neurons, so you can almost think of them like kind of like a connective tissue type of thing, although they are not a tissue type. But they do provide that support, that same function. They regulate nutrient ion concentrations and form this blood-brain barrier. That helps to just make sure that the chemical levels within the brain stay very, very level and very even, and can prevent entry of substances. The peripheral nervous system support cells are things like your Schwann cells that make the myelin sheath, and the satellite cells that to give this protective coating around the cell bodies. Here's the central nervous system. You can see those ependymal cells making the cerebrospinal fluid, astrocytes forming this barrier between blood vessel and the neuron, and making sure that just adding an extra level of filtration before things enter the blood, the central nervous system. Here's a microglial cell that's doing its phagocytic activities, and there's oligodendrocytes. You can see one cell is going to be myelinating multiple portions of a neuron. Versus in the peripheral nervous system, each myelin is a Schwann cell, versus having one oligodendrocyte making multiple sections of the myelin. Each one is an individual cell, and the satellites that are there to protect the neuron cell body. Neurons can regenerate. It takes a long time. They are the slowest of the regenerative tissues in our body. Your body is constantly repairing itself, whether there's actual damage or if it's just programmed death. And with the nervous system, it takes a full 7 years for your body to replace and replicate all those replicable cells. The rest of your body, your blood cells, red blood cells only last about 120 days. Every 3 months, you have a completely new set of blood cells. But it takes 7 years for your nervous system to regenerate. So every 7 years, you're a new person. Maybe do it sooner. That's up to you, right? So damaged peripheral nervous system neurons are able to regenerate their axons and then reestablish that electrical signal to that whatever that disconnected part of the body was. The neurolemma of the Schwann cells aids in this. It establishes this kind of route to be able to connect the axon to the original connection point. In the central nervous system, they are not able to regenerate. Okay, so when we talk about the nervous system, we've been talking about this electrical activity that neurons are conducting. And so we have to talk about charges inside of a cell. And so this is going to get into some electrical physics stuff. So just remembering that, we're talking about this electricity. So I always like to think about static shock, right? Like you're on a trampoline, because it seemed to always get shocked on a trampoline when I was a kid. And so you get a lot of static electricity, and sometimes you can almost see it. If it was so strong, you could see the little spark of electricity jumping from you to another person. That's what's happening with our neurons. And so when we're talking about this signal transduction, think about that. Think about some little shock being transferred from you to somebody else. So cell membranes of neurons have this polarity, and all of our cells have a polarity to them. But in the cells of the nervous system, they harness that polarity and change it to be able to establish this electrical conduction. So the inside of the cell membrane, the charge inside is different from the charge outside. It's more negative on the inside compared to the outside. And so that's the polarity. Think about the poles on a magnet. You have a north pole and a south pole, a positive and a negative end. That unequal distribution of ions allows, as well as proteins and things like that, allow the inside of the cell to be more negative than the outside. Neuron and muscle cells are excitable. This is that electrical, or rather they respond to that electrical change by changing their charge. Changing the charge then starts a sequence of events, and that's how neurons communicate. They send these electrical signals back and forth. So a lot of times with neurons, it's not when they say communicate, what they're really saying is that they're giving a yes signal or a no signal. It's not like, your neuron is going to tell you what Becky did at the pool party last week. That they're not communicating like that. They're just giving a, do more or do less, a yes, no, a stop, or a go signal. That's all that it is. It's very much like a binary code kind of thing. Ones and zeros, ons and offs. That's pretty much all it is. So when we talk about the polarity of a cell, we have to talk about the membrane potential. And this is the charge inside of a cell. That's the difference between the inside and the outside is the potential, because that's the potential difference. Resting membrane potential, for neurons, it's negative 70 millivolts. Depending on the cell in the body, it's going to be a little bit different. But in neurons, it's about negative 70 millivolts. As we said, it's the unequal distribution of both sodium and potassium ions. Those are really what regulate the nervous system function, specifically potassium. Potassium is very, very tightly regulated in our bodies. And then you have things like negatively charged ions and proteins that are going to be in cells. You shouldn't have proteins floating around outside of your cell. Proteins are going to be inside of the cells, and they're going to add a lot of negativity to that charge. So here you can use a voltmeter. You can see they're using a voltmeter. They have one inside the cytosol, inside the cell, and this extracellular fluid is outside of the cell. The purple are potassium, the pink are sodium, and the green, in this case, it's a chloride ion. You can see proteins inside the cell. And so this is a sodium-potassium pump, this membrane that's embedded in the phospholipid membrane bilayer. So you can see that it's exchanging 3 potassium for every 2 sodium. And it requires ATP. So this is this is facilitated, a facilitated action, meaning that it requires energy for it to happen. And that's because they're being moved across their membrane potential. You can see that on the outside there's already more potassium, and we're pumping 3 out. And there's more sodium in here, and we're pumping 2 in. And so it takes energy to move these against their concentration gradients, because naturally potassium is going to want to flow in, and sodium is going to want to flow out. So if you're having trouble remembering which side's negative, which side's positive, maybe that will help. Sodium and potassium are both positive ions, but for every 3 that go out, 2 sodium come in. And so that means that inside is negative relative to outside. So stimulus and the action potential. That we have talked about the resting membrane potential because it's that, again, potential is potential energy. It's both the difference in the charge, but also that's how much energy can then be generated by that neural cell. So neurons remain at rest until they're stimulated. They remain at that RMP of negative 70. And the stimulus can change it in either direction. It can make it more negative or more positive. You have an excitatory stimulus that opens chemically gated sodium channels. Those sodium ions flow in due to their concentration gradient, and the neuron becomes less negative. Remember, there's more sodium on the outside of the cell, and so, there's more sodium that's going to be stuck here on the inside of the cell, and so it will then make it less negative as you have more coming in. The threshold stimulus is a stimulus strong enough to cause so many sodium ions to enter that the potential changes from negative 70 to negative 55 to negative 55. So for a signal to be transmitted, it doesn't have to go from negative 70 to 0. Really, all that it needs is just to go from negative 70 to negative 55. You need that many sodium ions flowing inside to be able to make it that much less negative. Once it reaches threshold, then the voltage-gated sodium channel sodium channels open, and that's when you have a bunch of sodium rushing in, and it changes the charge to positive 30. And even though there are more sodium ions in here, the negative charge inside of the cell is what's outweighing that. So that's why you have sodium ions from the chemical and voltage-gated channels flowing in. Even though you do have more sodium ions inside the cell, this is it's that negative charge that's going to be pulling them in. And so notice that you have chemical-gated and voltage-gated sodium channels. So the chemical-gated channels, remember I said there's neurotransmitters that get released that then stimulate these neural cells. And so a neurotransmitter is going to bind to a neuron. It's going to open those chemical-gated sodium channels. And then once you reach threshold, the voltage-gated sodium channels turn on. And these are much more fast-acting than your chemical-gated. Voltage-gated electrical activity is much faster. And so then you start to have this action potential that gets created or began. And this is what travels down the axon, is the action potential. The change from negative to positive inside the cell is called depolarization, since now they're both positive relative to each other. So depolarization, so if something is polarized, it has a charge to it. If you're depolarizing it, you're removing that charge. So for a neuron to transmit a signal, it first has to reach threshold, right? So chemical gated channels, it has to be a strong enough, because chemical you can really regulate how much is being released and how much is not. And so you have to have enough chemical gated channels open to get to this -55. And then once threshold is reached, the voltage gated channel is open, because it's, again, that -55 millivolt charge. And that's when an action potential can be established. That then initiates this all or none response. So once there's an action potential, then that means it's going to be sent, it's going to continue fully down the neuron, or fully down the axon to the next neural structure. It either occurs or it does not. So an action potential will occur when the charge reaches -55 millivolts. So once the neuron reaches threshold, you are going to have a action potential. It's only right at that axon hillock, right at that place where the soma and the axon meet, that you have this kind of graded potential, where it's like that chemical gated one, where maybe it gets enough, maybe it doesn't. I sometimes think about it, that thing where you're kind of tired and all of a sudden your arm, like you go to kind of move your arm and it kind of moves, but it doesn't. It just kind of twitches a little bit, maybe. It's that. It's like you have like this little bit of signal, but not enough to really make the muscles move. But when you have an action potential, your arm fully moves. It jerks you awake, and you're fighting an invisible enemy or whatever it is. That's the action potential that will occur. And they're always the same strength. Action potentials of neuron are all the same strength. You're not going to have a weaker or less signal. You may have more or fewer neurons stimulated, but everyone that's stimulated is going to be giving a full signal strength. Once the action potential is reached, it kind of spikes and then it falls back down. And this process of returning to that -70, that resting membrane potential, is called repolarization. So it's recharging the neuron. Those are then established by the potassium and ions. So you have a lot of outward flow of potassium ions through those channels. Remember, it's 3 potassium ions moving out. And then that makes the inside of the cell more negative. Typically, there is this overshoot called hyperpolarization, where instead of being, because again, you're having this big rush outward of potassium, so it overcorrects, so it'll go down to -90, -110 millivolts, and then it establishes back at that -70. And then you have these sodium-potassium pumps reestablishing that concentration gradient of sodium and potassium. So here is a picture of that, and there's videos on your Connect website. This can be pretty confusing if you're having trouble picturing it. Watch some of those videos to try and understand this. So 3 phases. First, we have a cell at resting membrane potential. Then we have the depolarization phase, and then we have the repolarization phase. And so following these voltages, let's track the movement of the ions and these voltages. So first, you just have these membranes at rest. You can see all of the channels are closed. Nothing is flowing through. You have potassium inside, sodium outside. During depolarization, the sodium channels open. These are these chemical gated sodium channels, and sodium starts to flow in here, moving against that gradient. And then as that happens, you start to have the membrane potential changing. So it's starting to go more and more positive. So it's going from this resting, working its way up to threshold. And then once it reaches threshold, you're going to have this rapid depolarization. So the potassium channels open too, and potassium starts to flow out. Once it reaches +30, because again, these are voltage-gated sodium channels. So the voltage-gated sodium channels opened at -55, right here at that threshold at -55 millivolts. So, this is not meant to be cute, but just trying to state the obvious here. +30 is not the same as -55. And so once the voltage of the cell reaches +30, these voltage-gated sodium channels close, so that potassium can be the only thing diffusing out of the cell, which then hyperpolarizes it down here to -90 or so, and then levels back off at the -70. Here's just another depiction of that. Again, you can track the charges and very, very fast timing. Right, it's happening in just a few milliseconds. But remembering that you have the chemical gated sodium channels first, Then the chemical gated channels get you from resting membrane potential to threshold. From threshold, you have this voltage gated sodium channels open, and then at positive 30, the voltage gated sodium channels close, and the potassium gated channels are involved in repolarization as potassium rushes out and it becomes more negative again. So conducting this impulse, once the action potential has been triggered, it can then pass down to the adjacent regions of the axon. So remember, we talked about those nodes of Ranvier, those gaps in the myelin sheath where the signal will hop down. We call it a local current that allows it to flow from one region to the next. And that's that impulse conduction. So it's just passing it along. Like imagine, if you got shocked by somebody and then you could pass that shock on to somebody else. To ensure that it always moves in one direction and you don't have a signal going the wrong way, there's this thing called a refractory period. And this is a period of time after an action potential when that particular region cannot be re-stimulated. So it limits the frequency and it ensures a one-way direction down the axon. So here you can see that this part of the neuron, this part of the axon, this region, is positive. So this is what's been depolarized. The rest of the axon is sitting in its resting membrane potential state. But because this has been depolarized, this action potential has been stimulated, it's going to move down the axon. So it's going to shift to this next region. But as it shifts to this next region, this portion before it, immediately before it, is in its period of refraction. And so it's not able to depolarize again for a certain period of time, and that means that it's going to be continuing to go that way. Now, as this signal goes down, depending on what the stimulus is, let's say it's really bright lights, they just keep changing, going brighter, dimmer, like a strobe light thing. As soon as this signal has gone through and this first region has gone through that refractory period of hyperpolarizing and then going back to its resting membrane potential, it can then be re-stimulated again. So it may only, refraction is only a few milliseconds itself, but it's just enough time to make sure that the signal is going in one direction and then allowing the same area to be re-stimulated again. So here's just the steps of those written out if you want to read those. I'm not going to go reading through them because that's what they're there for. Okay, so types of impulse conduction then that we have, things like continuous, saltatory conductions. So continuous is in those unmyelinated axons. They do not have any of the myelin insulating it, and so the impulse has to go over the entire length of the membrane. Whereas saltatory means jumping. And so literally they're jumping, these are only in myelinated axons because they're jumping along those gaps within the myelin sheath. Speed then is also proportional to axon diameter. Thick myelinated go a lot faster than thin unmyelinated. You can see that's really no comparison between the speed. The thicker it is, the faster ions can flow through it, or the more charge can flow through it. And if it's myelinated, it's jumping. That's just going to make it faster. Here it is just, again, a depiction of that, showing it with a myelinated axon. So you can see that it's just these small little regions that are actually being depolarized. The cell doesn't have to depolarize within the Schwann portions. Okay, so a synapse then. This is where the neuron is meeting its structure that whatever it's communicating to. This is where they connect, right? So a synapse is a junction between two communicating neurons. There's always a small gap called the synaptic cleft, and that's what the impulse has to cross over to communicate. You have pre- and postsynaptic neurons. Presynaptic is sending, postsynaptic is receiving the signal. The cleft, they call that synaptic transmission, if you want to add that into your vocabulary. Communication, like I said, is used a neurotransmitter. Between the two electrical structures, you have those chemical gated sodium channels. They're going to be activated or deactivated by neurotransmitters. Neurotransmitters are stored in these vesicles, these synaptic vesicles. So a vesicle, remember, is just a little pouch that cells create, and this happens to be in the synaptic bulb, right, the end where the synapse is, so it's specifically a synaptic vesicle. It's not in the soma of the cell, right? And so that's where the vesicles will fuse with what we'll see is the knob, the membrane of the knob, to be released into the cleft. They then diffuse across that cleft and bind to receptors. So we looked at this in our lab last week, I believe, a little bit with the neuromuscular junction. It's a very, very similar structure. You can see you have the soma with its dendrites. The dendrites are for receiving. And so you have this axon right here at the synaptic knob, or right here at the synapse. You can see the synaptic knob, this enlarged portion. The cleft is the space between, and then this is representing the postsynaptic neuron. These are the synaptic vesicles filled with neurotransmitter. The little blue dots are the neurotransmitter. Of course, we have mitochondria. We've always got to have those. So as the electrical signal travels down the axon of the presynaptic neuron, I'm going to use a lot of technical terms here, so I'll try to speak slowly, but rewind and listen to it again if you need to. As the electrical signal travels down the axon of the presynaptic neuron, the membrane of the synaptic knob is filled with voltage-gated channels. And so as the voltage of the axon changes, of the presynaptic axon changes from that electrical signal, it's going to cause these vesicles to bind. And as they bind, they release their neurotransmitter into the gap. And then that neurotransmitter will then bind to receptors embedded on this postsynaptic membrane. Once these receptors, again, these are your chemical gated receptors, once enough of them start to bind this neurotransmitter, if there is enough to stimulate a response, then sodium will start to rush in, and you have that initiation of threshold reaching its action potential. This is just an electron micrograph, so you can see the same kind of structure. Those are mitochondria. These are all the vesicles, presynaptic knob, and postsynaptic membrane. So as I've been saying, there's excitatory and inhibitory neurotransmitters. Excitatory neurotransmitters increase sodium entry into the postsynaptic neuron. They bring it closer to threshold. There we go. They bring it closer to the threshold. Whereas inhibitory are going to increase the flow of those negative ions, chloride in or potassium out, to make the membrane even more negative, making action potential less likely. This sometimes this a lot happens like in vision in our eye receptors to let us know that it's more and more dark. We'll get an increase in the inhibitory neurotransmitters of those photoreceptors to and our body processes that as a darkening. Postsynaptic may have many presynaptic neurons influencing it, so it's the sums of the excitatory and inhibitory inputs of these neurons to derive its response. So as your body is getting, you go closer and closer to the central nervous system, you have more and more sensory receptors talking to fewer talking to fewer central nervous structures. And so a structure might be getting 4 or 5 signals, I'm just using that, it could be millions of signals, from the body, from the sensory receptors, but it has to take the aggregate of those. So overall, is it getting darker or is it getting lighter, right? Depending on which part of the room you might be focusing on, if there's other ambient light, you might have slightly brighter patches. And so you have this kind of cacophony of signals that our body then has to make sense out of. And so it takes whatever the overriding signal is, basically. This is just a depiction of excitatory versus inhibitory if you like to see that change in the voltage. So neurotransmitters, there are more than 100 neurotransmitters produced in synaptic knobs, stored in vesicles, things like acetylcholine, monoamines, amino acids, neuropeptides, estrogen and adrenaline and dopamine, serotonin, all of those are neurotransmitters. The action of the neurotransmitter, though, depends on the type of receptor. So you can have something like acetylcholine that will be excitatory in one neuron or inhibitory based on the kind of receptor that it has for that neurotransmitter. That just helps the body. It's easier to be able to, making 100 neurotransmitters is already hard enough. If you have 100 and then you can have different types of receptors, you don't have to then make 200 neurotransmitters or, 1,000 neurotransmitters. You can have a lot fewer neurotransmitters by just adjusting the receptor, because this is a lot easier to adjust. Some neurons will produce 1 while others produce 2 or 3. That's interesting. Here's a list of the neurotransmitters. You can go through these if you want to. I definitely recommend being familiar with at least the ones that are listed here: acetylcholine, norepinephrine, dopamine, serotonin, histamine. These are all and their basic actions. This will just help you if you're staying in medicine. You'll hear a lot about a lot of these. Amino acids, GABA. If you ever get put on gabapentin, or a lot of times this is a pain reliever and a what's the word I'm looking for? A sedative. Yeah. Okay. Releases of a neurotransmitter. These events just being again listed out for you, talking about the action potential coming down the axon to the synaptic knob. That then becoming more permeable is because of that change in the voltage, allowing the neurotransmitter to then be released into the synaptic cleft. Those then will fuse. Oh yeah, here we go. Sorry, I missed the calcium ion, allowing the synaptic vesicles to fuse the synaptic knob. Calcium ions are very helpful in that. So then neurotransmitter recycling. So after the neurotransmitter has acted on the postsynaptic cell, something has to happen. If that effect has to be stopped at some point, it may not be immediately, but after a while, again, going back to the vision example, it will get brighter, and so you need to be able to adapt to those changes. So there's destruction or removal of the neurotransmitter. It can be through an enzyme that breaks down that neurotransmitter. The common one is this acetylcholinesterase. Remember when you see this ACE esterase, that's something that's being broken down, and it will probably tell you at the beginning of the word what it's breaking down. Some neurotransmitters just travel back. They call it reuptake, like a little suction action where they just get drawn back into the cell. Drugs, things like cocaine, nicotine, methamphetamines, will inhibit these processes. Okay, how impulses then are processed. So again, as I said, it's a lot of yes or no signals, stop go signals, those excitatory, inhibitory, and it's a collection, a conglomerate of those signals. So you have neuronal pools. Neurons in the central nervous system organized together with varying numbers of cells depending on what they're supposed to be processing. It's receiving that input from the afferent neurons, or sensory neurons, and processing that with those interneurons. It can receive the excitatory and inhibitory input. If it's excitatory, but it does not sum to the threshold, there will be no nerve impulse. If it's excitatory and it reaches the threshold, then a nerve impulse occurs. So it can be one of those things where, yeah, there's a strong enough signal to, somewhat reach the nervous system, the central nervous system, but it's not really bright enough for us to actually need to start constricting the pupils again. We need to keep the pupils dilated because there's still not enough light to actually conduct a nerve impulse. Or somebody throws the lights on when you're in a dark room and you have this really big excitatory impulse. 3 processes that can happen: facilitation, convergence, and divergence. Facilitation, increasing the release of neurotransmitter in response to one impulse, which occurs in an excitatory presynaptic neuron upon repeated stimulation. So it increases likelihood that a postsynaptic neuron will reach threshold. So this is just making sure that facilitation is kind of aiding in stimulating a neuron. So you have the electrical and you have the chemical signal. Convergence is transmission of nerve impulses to a single neuron within a pool from 2 or more fibers. So this is kind of adding up those signals from separate neurons. And divergence is from a neural pool to several outputs to amplify an impulse. Here's just an example of a couple of those. You can see convergence, where you have 2 neurons coming onto one, and divergence, where one is then talking to 2, and then these 2 might then talk to 2 more, and so it's a much more amplified action. Okay, types of nerves then. A nerve is a bundle of fibers along the PNS. So it's going to look very, very similar to a muscle fiber in a muscle structure, as will a nerve. A nerve, though, is, again, a bundle of nerve fibers. Nerve fibers are your nerve cells. The types of nerves, you have sensory nerves. Again, those are the afferent going to the CNS. Motor, efferent, carry it from the CNS to effectors, and then mixed nerves that have both types of fibers. So a nerve, something like your cranial nerve or your vagus nerve or your facial nerve, is going to have different nerve fibers. They might be sensory fibers, they might be motor fibers, or they might be both. Most of them are both. A nerve has different tissue coverings to, again, establish that nesting doll structure that we saw in muscles. The epineurium, perineurium, and endoneurium. So this should sound very similar to epimysium, endomysium, epimysium, perimysium, endomysium, like we did for muscles. And even perineurium is surrounding fascicles. So here you can see an entire nerve that's wrapped in that epineurium. In between, you have this perineurium, which is defining your fascicles. And then each fascicle is going to be a bundle of nerve fibers. Then each nerve fiber is going to then be its own myelinated self. So the different neural pathways that nerves can take, the simplest of those is a reflex arc, and the most basic just involving 3 neurons. These are those involuntary reflexes that we have. The first thing is a sensory receptor detecting change. You have a sensory neuron carrying that from the receptor to the central nervous system, the interneuron processing it, the motor neuron taking it out, and then the effector, the muscle that carries out the reflex. So again, it can be very, very simple. You can have a receptor with its neuron taking it to an interneuron in the central nervous system, and then a motor effector output that can go all the way to the muscle. The knee reflex is one of those. You should definitely read through this and be familiar with the part, the description, and the function of the different structures within a reflex arc. So reflexes are automatic responses to changes inside or outside the body, helping to maintain homeostasis. So the knee-jerk one is one of those. That's a very simple reflex, but things like heart rate, blood pressure, your breathing reflexes, vomiting, swallowing, sneezing, all of those are reflexive actions. Again, the patellar one is the most basic, 2 neurons, a sensory and a motor. I'm sorry, it lacks in a neuron, doesn't even have one of those. So you strike the patellar ligament, that then sends it through a signal through the quadricep muscle. Sensory neurons transmitting that signal. Motor neurons transmit the impulse to the quadricep femoris, allowing them to contract. And there you go, you can see that being played out in this structure. This is one of those, I'm going to say this real quick, I think we'll get into it. This is considered a spinal reflex because this happens all within the spinal cord. It does not have to go from the sensory neuron all the way up the central nervous system to the brain and then back down the spinal cord and out the motor neuron. All of this takes place within the spinal cord. Our conscious thought of it, that happens in our spinal cord. So there's a branching in the sensory neuron. There's a branch in the sensory neuron that's then sending a signal for us to have conscious awareness of the sensation and the actions. But the actual sensory motor output happens all in the spinal cord, so it can be almost instantaneous. The withdrawal reflex is another one of those response to touching something painful. You have sensory neurons, interneurons, and motor neurons. Sensory neurons sending pain messages. There's those interneurons to transmit that information and figure out what kind of response needs to be had. And then motor signals to flex your muscles to contract. So there you can see that, if you step on something sharp, you have this interneuron, because it's a slightly more complex action, especially if you're trying to do something like withdrawal. That then requires a shifting of body weight, so you have to have other muscles on the other side of the body ready to balance you out. Okay, moving then into the meninges. So this is part of our central nervous system. I'm looking at the protective layers of the brain and the spinal cord. They're surrounded by these 3 membranes called meninges. You may have heard of people that have gotten meningitis before. That's an inflammation of these layers. They lie between the skull bones of the vertebrae and the soft tissues of the CNS. There are 3, the dura mater, which is the most external, arachnoid in the middle, and the pia mater, which is the most internal. The dura mater is the outermost layer, tough, dense connective tissue, a lot of blood vessels, it forms the internal periosteum of the skull bones. So this means that when you think of the dura mater, it's I'm trying to make sure I explain this correctly, or at least accurately. The brain and the skull, there's it's essentially glued to each other, but that's by these maters, these layers. And so the dura mater is technically the inside layer of the skull bones. It's not up against the brain, it's up against the inside of the skull bone. It forms partitions between the lobes, it forms dural sinuses for drainage, and it forms this epidural space, epidural, right? The arachnoid mater is the middle layer. It has this web-like layer, so that's why it gets this arachnoid, like a spider has this web-like kind of structure. And this is where you have the cerebrospinal fluid in the subarachnoid space. Going back real quick, epidural space. The reason we do epidurals there is because it has so many blood vessels. The pia is the innermost layer. It's actually the outer surface of the brain and spinal cord. It's directly attached to the brain and spinal cord. So here you can see the skin, then you have subcutaneous tissue, bone of the skull right in here. You have that outer compact, that inner spongy bone. And then you have the dura mater that line's just on the outside of it. You have this arachnoid mater with this web-like structures containing your cerebrospinal fluid, and then the pia mater right here, most internal. And there it is just on the spinal cord. Again, the spinal cord has the pia mater directly on top of it. Subarachnoid space, arachnoid layer is purple, and the dura mater is blue. All right, so let's look at the structures of the spinal cord. The spinal cord begins at the base of the brain, at the foramen magnum, so the big hole at the bottom of the skull, right below the brain stem. You have several enlargements, a cervical and a lumbar enlargement, or two, not several, but you have two enlargements. These are providing nerves to your upper limbs. The lumbar enlargement is giving the lower limbs. You also have this structure called the cauda equina, and this is where at the very base of the spinal cord where it starts to taper out, and it has a lot of these spinal nerves. So here you can see the brain stem at the top, and then as soon as you exit the foramen magnum, you have the spinal cord with the cervical enlargement, lumbar enlargement, and then the cauda equina. So your spinal cord has 31 segments, which connect to a pair of spinal nerves, so that can tell you how many spinal nerves there are. There are two grooves, the anterior median fissure and posterior median sulcus. Fissures and sulci are both names for the same type of feature. A sulcus is just bigger. So anterior median, that's your location. Fissure is your structure. This helps to divide the cord into right and left halves. You also have white matter versus gray matter. You probably have heard of that related to your brain. Oh, the white matter, the gray matter, blah, blah, blah. White matter is fatty tissue. That's the myelinated nerves. Gray matter is interneurons. Those are unmyelinated. That's really what you can kind of remember. White matter, myelinated. Those are tracts carrying signals. Gray matter, unmyelinated interneurons, processing signals. Cell bodies, sensory neurons are in the posterior root ganglia, so the actual spinal cord only contains axons. It doesn't contain any cell bodies. And there are also things called the horns, the posterior and anterior horn and a lateral horn. These are just portions of gray matter. Gray matter, those horns, divide the white matter into these funiculi, these columns, anterior, lateral, and posterior, each having bundles of axons called tracts. So this is, again, just allowing the spinal cord to be able to determine where information needs to go and how it should be processed. It's like a highway system. You can't just have complete chaos or things get really shut down very quickly. It has to be very efficient where information is going and how it is getting there. And then there's a central canal in the middle of the gray matter that actually has the cerebrospinal fluid. So here you can see all of those features, this external white matter, the internal deeper gray matter. The gray matter has its horns, and then you have the different columns of the white matter. And then these are the fissures and the sulcus. So function of the spinal cord, transmitting impulses to and from the brain, and housing the spinal reflexes. So there's reflexes that happen here, but there's no higher order thinking that happens in the spinal cord, no real autonomic processes that happen there. The spinal cord has ascending and descending tracts. Obviously, ascending is carrying sensory information to the brain, whereas descending is carrying motor signals to the muscles and glands. Okay, so there's just another way for you to arrange it. You have afferent sensory neurons on ascending tract signals and descending motor neurons that are efferent signals. Names identify the nerve tracts, the origin and termination of the fibers. Spinothalamic, corticospinal, extrapyramidal. So this is telling you where it's coming from and where it's going to. So this one is from the spinal cord to the thalamus. This is a motor impulse from the cortex of the brain to the spinal cord. And the extrapyramidal involved with balance and posture. So this is a good thing to kind of add into as you're studying, if you have the ability to add that in. And as I talked about spinal reflexes, those are just passing through the spinal cord. They don't have to go up and down these tracks. Even though the sensory information, the cognitive awareness of it, will have to go up a track. Here you can see one of those tracks being passed through. And then descending tracks coming back down with motor signals. Okay, so the brain. Brain is the largest, most complex portion of the nervous system, has 100 billion multipolar neurons, a bunch of neuroglia to support all of that, and its structure is reversed from the spinal cord. In the brain, the gray matter is on the outside, white matter is on the inside. Okay, gray matter, unmyelinated interneurons on the outside, white matter, those receiving signals from the spinal cord into the white matter, up those tracks, and then moving it through the brain. So that's all myelinated. The brain is divided into 4 major regions. Cerebrum, that's what you think of, like when you think probably of your brain, you're thinking of the cerebrum that has all those folds and the lobes. It has higher mental thinking, sensory and motor functions. The diencephalon is deeper inside the brain. It has a lot of sensory input, a lot of homeostatic or autonomic processes, blood pressure, not blood pressure, memories, emotions, those types of things. Cerebellum, this is at the very bottom of your brain. This is very muscular activity and motor coordination. And then the brainstem regulates visceral activities. This is where your blood pressure, things like your respiratory rate, swallowing reflexes, all of that is in your brainstem. Here you can see the cerebrum is in gray here with all of its folds. Then you have the diencephalon, which is here marked by this corpus callosum. You have the thalamus, right? The pineal gland, the pituitary gland, all of that is within the diencephalon. You have the cerebellum back here. This is the little brain, so cerebrum, and then the cerebellum down here. And then you have your brain stem. It has 3 regions: the midbrain, just inferior to the diencephalon, that thalamic region, the midbrain, the pons, and the medulla oblongata. And then just after the medulla oblongata, it's continuous with the spinal cord. Here's a dissected version of that. Okay, looking at the cerebrum, I'm going to go through this probably fairly quickly because we're going to cover it in lab as well, but I want to let you have this video. So the cerebrum has 2 hemispheres, the left and the right side. And then it has the corpus callosum that attaches those 2, that allows the 2 to talk to each other. And yes, the right side of your body is controlled by the left half of your brain. The left side of your body is controlled by the right half. There's this, we call it a desiccation of can't remember the word, but it does switch as the sensory and motor neurons, or as the sensory neurons are coming in. Right is left, goes to the right. But you use both sides of your brain, and let me go ahead and also say you use 100% of your brain. It's not like you only use 10% of your brain. You use 100% of your brain. It would be very bad if you only used 10%. You would not be conscious. So the corpus callosum helps to connect those 2 sides, so that way our body always knows what's happening on each side. There are surface features. There are things called gyri, singular is a gyrus, and that's a ridge, whereas a sulcus is a groove, and then a fissure is a deep groove. Okay, so the bumps that you see on the brain are gyri. The grooves, the small ones, are sulci, and then there's deeper ones called fissures. There are 4 lobes that correspond to the bone that they're underneath: frontal, parietal, temporal, and occipital. You should know where those are by now. There's also a 5th lobe called the insula that is deep to your temporal lobe. There's a thin layer of gray matter in the cerebral cortex, just outside of the cerebrum, and that has 75% of the cell bodies in the nervous system. Okay, so think about it, 75% of the cell bodies and this thin layer of gray matter. Beneath that, beneath the cortex, you have the white matter that makes up those myelinated fibers. So here you can kind of see the different regions, the different lobes highlighted, as well as certain specific areas where things are processed. Wernicke's and Broca's areas are for speech. You have the insula, this is a lot of times for taste. Frontal lobe is things like higher order thinking, decision making. These 2 areas right here, this one that's pink and this one that's yellow, you should definitely be familiar with these. So you have the central sulcus right here, and then you have just in front of it this primary motor cortex and the primary somatosensory cortex. And those are different regions where you have motor output and sensory input. And so those are big areas that you should be familiar with. Here's a human brain. Okay, the cerebrum, breaking down its functions in lobes. So interpreting sensory input, initiating voluntary muscle movements, storing information for memory, integrating information for reasoning, intelligence, and personality. All of this is happening in the cerebrum. Functional areas of the brain do overlap, but it can generally be divided into sensory, association, and motor areas. Association is the higher thinking, the putting things together, interpreting signals. Sensory areas are located in several parts of the cerebrum. You have cutaneous, visual, auditory, taste, and smell areas. And you can see they're located in particular parts of the lobe. So if you were to have damage, stroke, let's say, in the posterior occipital lobe, that could then impact your visual acuity, but you would still be able to taste, smell, and hear just fine. But it's just your vision that's impaired. And as I said, they cross over, so right side is left, interpreted by centers in the left hemisphere, and vice versa. You should be familiar with just generally, it can be pretty tough to start to break down all where everything in the brain is processed. So I don't expect you to know everything, but generally you should be able to understand where at least the 4 main regions, right? What each one is providing, what each one is doing. If you don't know if you have trouble remembering where each sensory area is, that's not as important as being able to know what the different lobes do. Okay, so with the cerebral cortex, you also have the 4 lobes of the brain, or 5 lobes of the brain if you include the insula. And this is where things like that higher order reasoning, judgment, emotions, verbalizing ideas are all stored. So yes, I would expect you to know that the frontal lobe is higher intellectual processes, planning and problem solving. Parietal lobe, speech. Occipital lobe, visual patterns and sensory information. And then it does not have the insula on this page. So yeah, be familiar with all of those. Primary motor areas, as I was saying, that primary motor cortex in the posterior frontal lobes anterior to the sulcus. So posterior frontal lobe, but anterior to the central sulcus. There are also pyramidal cells in there that are helping to connect spinal cord and skeletal muscles, and that crossover happens again. Broca's speech area in the frontal lobe on the left side controls muscle movements for speech. So that's why you have different things for speech. So Broca's area is the muscle movement. Wernicke's area, as we're looking at in this one, it's understanding written and spoken language. So perhaps you have all the ability to say words and you understand what people are saying, but when you actually speak, you just speak gibberish. It sounds like gibberish because your brain doesn't know how to pick the correct word. You say it just fine. You understand what people are saying. You know what you want to say, but when you actually speak, rather than speaking normal or speaking coherently, it would just be a random string of words. and potentially same thing for writing them. The brain is very, again, there are segmented areas where things are processed, but there's so much overlap, it's really difficult to start trying to interpret where everything gets processed because it's so open-ended. Hemisphere dominance. Both cerebral hemispheres receive and analyze sensory input, but one side is the dominant hemisphere in most people. So again, you might have some more predilection to left hemisphere things such as speech and writing, whereas right hemisphere might be more for spatial awareness and reasoning and those kind of things. But you use both sides. It's a lot of this is determined by the structure of your neurons, and so when you practice one thing, you can get better at that. You will create more neurons. And really, the corpus callosum is connecting those two. And so in females of our species, estrogen causes a thicker corpus callosum. And so females tend to have more equal dominance versus one being more dominant than another. All right, so the basal nuclei are masses of gray matter deep within the cerebral hemisphere. Also, you'll hear them called the basal ganglia. They have these different structures within them, and they produce dopamine, help control motor activities, facilitate voluntary movement, and then they can be signs of other diseases, Parkinson's and Huntington's disease. This is showing where those basal nuclei are located deep within the cerebrum. And then you have ventricles. Ventricles are cavities that are helping to drain or circulate and drain cerebrospinal fluid. Two lateral ventricles, interventricular foramina, which are small holes between the ventricles, what they call the third ventricle, and the fourth ventricle. An aqueduct, a cerebral aqueduct, is a storage area. So you can see there are these kind of big loping things. Don't get them confused with the corpus callosum, right? It kind of looks slightly similar shape. These are for circulating cerebrospinal fluid throughout the brain. A choroid plexus. A plexus is a network of nerves. Whenever you see plexus, it's just a network of nerves. The choroid plexus are masses containing specialized capillaries from the pia mater. They secrete the cerebrospinal fluid into the ventricles, and it circulates through there into the subarachnoid space and then reabsorbed back into the blood. So that way it doesn't just build up in the brain. CSF completely surrounds the brain and spinal cord. It has nutritive properties as well as protective cushionings. Protective functionings, rather. Protective cushion functions. Ugh, my mouth doesn't want to work that word. So it's going to aid in nutrient exchange, things like glucose, oxygen, as well as removing carbon dioxide and waste from the nervous system. You can see how cerebrospinal fluid is circulated through and then up and out of these little choroid plexuses, that variation of the pia mater, and then moved into the ventricles where it can then be drained out. This to make sure that the cerebrospinal fluid doesn't swell up in the brain and put pressure on the brain tissue. The diencephalon is between the cerebral hemispheres above the midbrain, so just above the midbrain of the brain stem. Consists of mainly gray matter. It has the thalamus and hypothalamus. There's also things like your optic tracts, your infundibulum, which is the attachment of the pituitary gland, mammillary bodies, and pineal gland as well. So important structures are in the diencephalon. Thalamus is for directing, sorting sensory information coming from different parts of the nervous system to the cerebral cortex, channeling all sensory impulses except for smell, and general awareness of the sensations such as pain, touch, temperature. So just making sure that we're actually aware of what we're experiencing. The hypothalamus maintains homeostasis with a lot of, by linking the endocrine system with the nervous system. So when we get into the endocrine system, we'll learn more about the hypothalamus because it is kind of the governor of the endocrine system. You can read through its functions right here, but a lot of times it secretes hormones that then will have effects on other structures. It's not like your endocrine system, your hypothalamus releases hormones and they go directly into the bloodstream. A lot of times they are then impacting other structures, which then will take over. Particularly your pituitary gland. Your hypothalamus regulates a lot of the actions in your pituitary gland. The limbic system, this is part of the diencephalon, emotional experiences and expression. So it has different parts to it. Modifies behavior, producing feelings of fear, anger, and pleasure. So your amygdala, right, is in here too as well as pleasant or unpleasant feelings. Yeah. The brainstem, as we said, it has three parts: the midbrain, pons, and medulla oblongata. Right at the base of the cerebrum, but still superior to the brainstem, connecting these three areas to the spinal cord. When we get into the individual systems, the cardiovascular and respiratory systems, we'll see in more detail what roles these areas play. Here you can see the brainstem, those areas of the brainstem. Midbrain, bundles of nerve fibers conveying impulses from higher centers to and from the brain. Visual and auditory reflexes and pathways between the cerebrum and lower portions of the nervous system. The pons transmits impulses to and from the medulla oblongata and the cerebrum, as well as regulating rate and depth of breathing. The medulla oblongata stays real busy. It is helping to transmit information, again, from the upper to lower nervous system portions, and it has cardiac center, vasomotor center, and the respiratory center, all which do what they sound like, regulating heart rate, vasoconstriction and dilation, blood pressure regulation, as well as breathing and your respiratory rates, with reflexes for coughing, sneezing, swallowing, and vomiting. It's a busy area, but it also makes sense to have all of this in one spot. So that way your breathing is regulated with your swallowing and sneezing. As we get older, we don't have to think so much about breathing while we're eating. When we're younger, it's much more dangerous because this part of the body is not fully developed. And so it's a lot easier for infants to choke because their medulla oblongata, these reflexes, aren't as well established as they are as we get older. You also have reticular formations. These are networks of nerve fibers connecting small masses of gray matter throughout the brainstem. They connect pairs of the hypothalamus, basal nuclei, cerebellum, and cerebrum. Decreased activity results in sleep, and increased results in wakefulness. There you go. The cerebellum is beneath the occipital lobe. So again, talking about the four main regions of the brain, we've done the cerebrum, diencephalon, brainstem, and now we're looking at the cerebellum. This is located right at the base of the brain, at the posterior portion. It has two hemispheres as well, so it looks like a little brain, pretty much. Whenever it's cut open, you see this arbor vitae structure, this tree of life, if you want to call it that. It communicates through other parts of the central nervous system through three different tracts, the cerebellar peduncles, and it is necessary to integrate sensory information about the position of the body parts. So your proprioception is what that's called, your awareness of your body in space, as well as coordinated muscle activity, posture, and movement occurs in the desired manner. So any kind of skilled movement that you might have. I'm a crocheter. I've been crocheting for about 12 years now, and so some of those actions are just ingrained in my brain now. And so that's a very skilled action that my body doesn't have to think about so much because it's stored in the cerebellum. When you are intoxicated, this area is incredibly affected, and so that's why they do things like field sobriety tests. Not knowing how to put your finger to your nose is a function of the disruption of nerve signals in the cerebellum. Same thing like not being able to walk a straight line. There you can see the cerebellum down here with its arbor vitae. Okay, let's get into the peripheral nervous system then. These are nerves that connect the CNS to body parts. You have cranial and spinal nerves, and sensory and motor divisions within that. There are two portions of the peripheral nervous system. The somatic nervous system, connecting central nervous system to skeletal muscles, skin, and oversees conscious activities, as well as the autonomic nervous system, which is all subconscious stuff, and your organs. So you should be pretty familiar, very familiar at least, with the different subdivisions of the nervous system. So read through this, make your own little charts if you need to. Okay, looking at the cranial nerves. I'm not going to require you for this course to memorize the names and numbers of the cranial nerves, but you should. It may come up on questions, right? It may happen. I'll try to limit that as I can, but yeah, I'm not going to use it in lab. I'm not going to make you identify the cranial nerves. So 12 pairs of cranial nerves from the underside of the brain. Most of them are mixed, meaning that they have sensory and motor fibers, but some are exclusive. They are designated by number and name, and they are in order for the most part. So when you look at the brain, you have all the way at the very front, cranial nerve 1, your olfactory bulb, and then as you go back, you have the optic, which is cranial nerve 2, oculomotor, moving the eyes is 3, trochlear is 4, and so on and so forth. I said I'm not going to make you learn them, but for identifying, but there are some that you're just going to have to be familiar with. Thankfully, a lot of them, a few of them are fairly obviously named, but some things like your vagus nerve, that's just a very it's a strong parasympathetic nerve that's involved in autonomic regulation. So it strongly regulates a lot of your body processes, your internal organ processes. And so some of these you're going to be hearing a lot. Here is those nerve names, numbers, types, and functions. Again, you might get questions on these, so be prepared for that. But it's a lot to try and learn these nerves sometimes, on top of everything else in the nervous system. I recognize that. The spinal nerves, 31 pairs of spinal nerves, all except the first are mixed. So that tells you just that first pair is mixed. Everything else is just that first pair is not. Everything else are mixed. Numbered in sequence: 8 cervical, 12 thoracic, 5 lumbar, and 5 sacral, with 1 pair of coccygeal nerves. There are 2 roots coming off of each spinal nerve. They have a posterior and a motor anterior root. And each posterior root has the posterior root ganglion, because remember, when we're talking about the spinal cord, the bodies of the cell neurons are outside of the spinal cord. And so they're in that posterior root ganglion. Ganglion is a word for a collection of cell bodies. The anterior and posterior roots unite to form a spinal nerve that then extends out through the vertebral canal. So you have 2 roots coming off. You have, there we go. You have your spinal cord that runs down your body, and then off of the spinal cord you have 2 roots. You have an anterior and a posterior root that fuse to form the spinal nerve carrying sensory and motor information. Sensory information comes in through the anterior root, and then motor information goes out through the posterior root. And then you have the ganglion, which is this line of cell bodies on the outside of the spinal cord. There are several plexuses throughout the body. Again, a plexus is a network. The names will tell you where they are. Cervical plexus is in the neck, controlling things like the phrenic nerve, which is regulating your diaphragm. Your brachial plexus, brachium to the upper arm, so skin of the arms, forearms, and hands. If you get nerve tingling in your fingers, a lot of times it's from this brachial plexus. Well, depending on where you're getting the tingling, it could be cervical or brachial plexus nerves. And the lumbosacral plexus is in the low back of the spinal cord. Muscles of the skin, lower abdomen, genitalia, your reproductive and urinary functions, as well as your legs, and the sciatic nerve. Anterior branches from the thoracic spinal nerves also become your intercostal nerves for in between your ribs. And they serve to sort and recombine spinal nerve axons. So axons derived from the spinal nerves extend to the same part of the body in the same peripheral nerve. It's an organizational thing. That's really what it comes down to. Okay, looking at the autonomic nervous system, and then we'll get into the somatic nervous system. So the autonomic nervous system is part of your peripheral nervous system, functions constantly and independently without conscious effort. You don't have to think about moving or the release of digestive enzymes into your small intestine or your body making bile. It automatically does that thanks to the autonomic nervous system. So those are the visceral motor functions, right? Visceral meaning your glands in your body. Smooth muscle, cardiac muscle, glands, helps to maintain homeostasis, responding to emotional stress, prepares the body for strenuous activity, so fight or flight, rest and digest, that kind of stuff. And then controls heart rate, blood pressure, breathing rate, and body temperature. So autonomic nervous system keeps you going, keeps you moving. Autonomic activities regulated by reflexes that have receptors in your organs and your skin. Impulses conducted to the brain or spinal cord. Motor impulses travel through the nerves and then to the effectors. And there are 2 divisions of the autonomic nervous system. Sympathetic, fight or flight, So during stress or emergency, you're having sympathetic stimulation, and parasympathetic, under normal restful conditions, rest and digest. So it kind of tells you what they're preparing you for. So in your autonomic neurons, they're all motor neurons, and they consist of two pathways. Preganglionic leaves the central nervous system, synapses with one or more, and they have cell bodies and an autonomic ganglion, or the PNS. And the postganglionic, which leaves an autonomic ganglion and innervates a visceral effector. So essentially it's this extra string of cell bodies that you have running along this spinal cord. And so you can see this preganglionic, this autonomic ganglion here. You have the spinal cord, you have a spinal nerve, right? You have a posterior root, this is your motor signal. You have an anterior root, which is going to be your sensory signal. I'm sorry, I did that wrong. Yeah, no, I did it right. No, I did it wrong. Posterior root with its sensory, anterior root with its motor signal, and then as this motor output goes, it has to synapse with this autonomic ganglion, and then the postganglionic fiber. So this is what's going to be regulating that autonomic response. Versus somatic, where you don't have this chain, this ganglionic chain. You can just have the posterior sensory and anterior motor root going to your skeletal muscle. So in the sympathetic division, you have short preganglionic fibers coming from the thoracic and lumbar regions that go to the ventral roots of the spinal nerve, ventral meaning to the front, right? Ventral versus dorsal. The axons then leave spinal nerves and proceed to the sympathetic ganglia, a chain of sympathetic cell bodies right beside the vertebral column. They synapse with postganglionic neurons. They have long axons that then proceed to visceral effector. So short preganglionic in the sympathetic, long postganglionic. Yeah. Here you can kind of see that fibers and the structures that they're going to. This portion lower down in the spinal cord, remember it's lumbosacral regions, and the sympathetic chain ganglia, right? So sympathetic referring to the autonomic nervous system, and ganglia being those cell bodies. And then from the short preganglionic, you have these long postganglionic. So just along the the side of the spinal cord, you have nerves that run all the way down to your digestive organs, your reproductive organs, those things. Parasympathetic have long preganglionic in the parasympathetic division, and they come from the brainstem and the sacral region. So very much superior and slightly inferior to the sympathetic division. Preganglionic fibers extend out and synapse in terminal ganglia close to the visceral effector organ with short postganglionic fibers, so the opposite of what the sympathetic has. So you can see these long preganglionic fibers going to the organs, and then they have a short postganglionic that they synapse with. And again, based on location, up here in the brainstem, down here in the sacral plexus. So autonomic neurotransmitters, we can talk about preganglionic fibers, the sympathetic and parasympathetic divisions, all release acetylcholine. These are cholinergic fibers. Most of them, the sympathetic, are going to release norepinephrine. These are adrenergic. Noradrenaline, they're adrenergic fibers. So sympathetic is getting you ready for that fight or flight. So norepinephrine, epinephrine, adrenaline, adrenergic. The effects are different due to those effects of the transmitters, acetylcholine versus norepinephrine. And usually they have opposing effects on organs. So most organs are going to receive innervation from both divisions. It's not completely true. Sometimes they're only affected by sympathetic or parasympathetic. But those ones that do have dual innervation, they're going to be opposing ones. And so it's whatever signal is the most dominant. Here you can just see a visual difference between parasympathetic and sympathetic. Sympathetic have this short preganglionic chain. Parasympathetic have a long preganglionic fiber. Parasympathetic are using acetylcholine in this sense. And then the sympathetic, they can also use acetylcholine, but then they'll also be releasing norepinephrine. Here are the effects of or the visual effector, so pupil of the eye, heart rate, bronchioles of the lungs, in response to sympathetic or parasympathetic stimulation. You should know these. So you should know what happens when the muscle of the intestinal wall gets sympathetic stimulation. It's going to slow the peristaltic action. So it's going to sympathetic stimulation is going to slow the digestive system, whereas parasympathetic is going to increase the digestive system. Same thing. So a lot of this, you have to remember, the sympathetic is fight or flight. So it's about using your resources to help you get away from a dangerous situation. So the pupil of the eye, when you have sympathetic stimulation, is going to dilate, so you can take in more light. Versus parasympathetic, it will constrict because that extra light is going to be stimulating and keep your brain aware. And you don't need to be aware if you have parasympathetic stimulation. Heart rate is going to increase from sympathetic because that's going to increase your blood pressure and your delivery of nutrients, whereas in parasympathetic it can decrease. Bronchioles of the lung are going to dilate so you can take in more air versus constrict with parasympathetic because you don't need that extra energy. And so on and so forth. You should go through all of those. So the autonomic nervous system is mostly controlled by the centers in the brain and spinal cord, things like the limbic system and cerebral cortex, alter reactions of the autonomic nervous system through emotional influence. When you are upset and scared versus when you are maybe, let's think nervous, right? So if you're nervous about something, you might start sweating. It changes your body temperature. That response to emotions is your limbic system and your autonomic nervous system talking to each other. Okay, hope you enjoyed that lecture. Thank you very much.